One. Hello, my name is William Brady Diaz,
And I'm Christopher Church.
and we're going to be talking about our capstone, which is RNA Motif Contrast Web Service, and our mentor is Dr. Islam.
All right. So some context to know what we've been working with is our project has to do with the study of RNA molecules. An RNA molecule is found essentially in all living organisms on Earth. And what's special about them is that they can fold in space or twist in space into certain conformations called motifs. So a motif, if we imagine like an RNA by analogy as like a string or a thread, if you like twist a string into like a knot, you tie it up into a knot, that knot is the motif. Um, and similarly, you can have some motifs that are more similar to others, since you can have some knots that are more similar to others based on how they were tied. And that process of ranking different RNA motifs by similarity is called clustering. And visually, I have images here showing that process. So at the bottom right of the screen, here are some RNA molecules that are just normal, unanalyzed, but they each have portions of their structure that are similar to one another. And if we look above it, the motifs have been colored. So we can see that there's like a green colored motif, those are similar, and there's a blue colored motif, those are similar. And the reason why this is important is because the structure of these RNA molecules influences to a great extent their biological function, like what proteins are going to be produced and so forth. In fact, there are vaccines that rely on RNA molecules themselves, and this would be an application of that. And to study motifs, a variety of tools have been produced. Our mentor, Dr. Islam, has produced one called RNA Motif Contrast, and we'll explain a little bit about that right now.
Yeah, so like Chris was saying, our mentor has created a tool called RNA Motif Contrast, which is used to detect subfamilies from a list of motifs. But there's current limitations that they have, such as it requires a local setup. There's also some license constraints with the rendering of the mesh files that's used for the output and the static images. It also requires a very extensive command line expertise. And like I said before, it only produces a static output that's a photo. But with our web service, what we're going to be able to do is provide researchers a browser-based workflow that can be accessed from anywhere with an internet connection. Like I said, in cloud execution. So under-resourced labs will have access to this powerful tool that they wouldn't have had access to before. We're going to be able to provide interactive 3D results as well. So researchers can interact with the motifs and tag them and things like that. And then as well as saved analysis, which is going to be able to facilitate a collaborative workflow among different researchers in the field.
Okay, so to reiterate from that, the revisions we're adding to the current RNA motif contrast is going to have the three following impacts. One is just going to be more precise as a web service. We already mentioned that we're implementing a GUI to allow a more intuitive manipulation of it. That's otherwise not possible. You can now color code parts of a motif and see that live in action as you're color coding it. There's some images there on the right that would show what that might look like. Definitely better than looking at text on a command line. Second, it would be more accessible as a web service because there's a lower barrier of entry. You don't need to know how a command line workflow is meant to function. You don't need to know programming languages as much. Ultimately, it is better for our demographic of interest, which is biologists and perhaps students that just aren't as CS savvy. Lastly, it's more collaborative because it allows saving you can upload your data sets, color code them or tag them as we call them, group your molecules based on similarities, and then save it. And you can make that data set public for anyone or keep it private within an organization and share it with other researchers, or perhaps even allow real-time multiple users at once editing the single data set at the same time, regardless of where they are in the world.
Yeah, and then so to facilitate this workflow, we utilized a microservice architecture. And how it works is we have our front end, which is built using Next.js, React, and Babylon.js. And the front end is where the users are able to interact with the motifs, save their analysis and things like that. And then we have our Nest.js API, which handles all the jobs that are going to be created, the authentication logic, and then saving sessions and things like that. And then the reason why we have jobs is because some of the workflows are very computationally expensive, especially when there's a large list of motifs. And we implemented a producer worker pattern or worker queue pattern that allows us to do asynchronous workflows, so that way our API doesn't get bottlenecked. And then that leads into our Redis and BullMQ instance. And this is how our queue is being managed. So our backend API is pushing jobs onto this queue. And then from there, we're able to, like I said, have an asynchronous workflow. And then our next two microservices are Taiga, which is used for generating the CIF files. And these contain the atom information and detailed information about different motifs that the user may have uploaded. And then we have Tundra, which is used to generate the mesh files the user is able to actually interact with. And these mesh files are used to render the 3D motif that in the front end. And then we have our Supabase instance, which is used for storage and caching. We have the motifs, the CIF generation, and the mesh files that are generated being saved in our Supabase. So that way we don't have to keep generating new mesh files and CIF files over and over again. We just check to see if they're already cached, and then it saves some time in the workflow. And then finally, we have our Python worker, and then this is where the main RNA motif contrast program is. And then what it does is it listens to our queue, and then when a job is added to the queue, then our Python worker picks it up, and then it begins to execute that job. And then once it's done executing, it sends it back to our server, a Redis server, and then it lets the API know, and then it alerts the user, and then from there we're able to inspect all of the output generated by RNA motif contrast.
Mhm.
And then, yeah, so how it works is the user uploads a file. It can be a text file, CSV file with motifs. Then they run the program, and then through after it goes through our pipeline, they're able to explore the motifs, different subfamilies, things like that. And then, like we said, they can also save their analysis. And then basically, we've created a pipeline that's able to facilitate a collaborative research workflow.
Let's just start the demo.
And then, yeah, so now we have the demo, and this is a screenshot, and then I'll let Chris take over for the demo.
You guys see some of them? Yeah, I got it. So, let's go back to the homepage quickly. So, yeah, here's the homepage. Normally, the user, when coming in, would be able to select a file here and upload a text file with the motif data that they want. However, it takes a while to render this. So instead, we're going to go to the user dashboard and look at a previously saved data set. So we get to show how that works. And oh, look, it's saved. How nice and convenient. So the first thing to notice is at the top left, we have a layers panel. That's going to have a bunch of objects in it. In this case, objects is a structure, a motif structure. So you can select them based off of here. Like now I've just selected this one and rotate it. But one thing we can see is each one has a variety of colors or tags. Like this one has a red one, a magenta one, and a cyan colored one. These tags are not just arbitrary. If we go to like this tag menu, each one represents some kind of biological feature in the motif. So supposedly, according to this, the red tag would be a kissing loop. Got it. And if I felt that another structure, like this one down here, also had a kissing loop, then I could color it if I so wished, and then begin doing manipulations based off of that. For example, if we looked at the analysis panel, if we selected, let me give a second. If we selected more than one, It would begin producing an RMSD score to show how structurally similar they are. And we can do that based off of the tags as well that we just mentioned. So if we were only interested in getting the similarities based off of the kissing loop, then I would click that and it would tell me that they have an 8.85 similarity, the red in the first structure with the red in the bottom structure. So roughly that just shows how the user might be able to use this and orient the molecules based off of similarity. So this is apparently a better way of rotating it in space to help the red regions overlap. And that's how the user would be using it.
And then yeah, so another important feature that we have too is the labeling. You can see the actual indices within the structure, and then this can be used for, advanced analysis and things like that. And then yeah,
Yeah.
so what we've done is we have turned a prototype project into a fully functional research workflow that can be used by researchers around the world. And yeah, we just want to say thank you to Dr. Islam and then other RNA 3DS lab members who supported the initial development. Yeah, thank you.
No, thank you. Appreciate it. Okay, so do you think we should upload this?